I'd now like to introduce our first speaker, William Dynan, who earned his Bachelor of Science degree from Massachusetts Institute of Technology and his PhD from University of Wisconsin. His postdoctoral fellowship was at the University of California at Berkeley, and where he discovered the first human promoter-specific transcription factor, that is SP1. He's currently a professor and Georgiaville Research Alliance Eminent Scholar at the Medical College of Georgia. And we'd now like to welcome Dr. Dynan to the podium to give his talk. Thanks. Thanks so much.
Okay. I don't know if you can get the top of my head with that camera. So I want to thank the organizers very much for inviting me to such a diverse and interesting meeting. My topic for today is understanding and re-engineering nucleoprotein machines to cure human disease. It was carried out in the Nanomedicine Center for Nucleoprotein Machines, which is a consortium of universities in the US and Europe, funded by the NIH Roadmap for Biomedical Research. So nanomachines, natural nanomachines, they fall into four categories: those that generate mechanical force, those that generate chemical gradients, those that carry out chemical transformations, and finally those that carry out information storage and transfer. And nucleoprotein machines are concerned with the last of these functions, fundamental functions: information storage and transfer. They are characterized by an ability to self-assemble within the cell nucleus. They carry out processes of DNA replication, copying of DNA into RNA, translation into processing of RNAs. The particular machine that we work with is the machine that's concerned with the repair of DNA double-strand breaks, and I'll talk about those more in a moment. But first, my theme for today is the development of simple nanodevices, that is, devices with one or two components that interfere or give new functions to these complex nucleoprotein machines. So this concept's a little different than what Ralph Merkel, for example, talked about yesterday, which is this sort of bottom-up engineering of machines with new functions according to a complete design. And it's more in the nature of working with this evolutionary process of biology that we just heard about in Dr. Bloomberg's talk. The idea that cells really have a lot of parts lying around that can be adapted for new purposes. There are all kinds of examples of this in biology, and I'll give you an example at the end of my talk today. The particular machines that we work with are those that repair DNA double-strand breaks, and I'm going to give just three examples today of what we're doing with these. First, we're modifying the machines in a way that we can see them in the optical microscope by introducing bright photostable probes into components of the machines. Second, we've developed a way to introduce a modified single-chain antibody into cells to inhibit the function of nucleoprotein machines. And third, we're working on a way to introduce new functions for gene correction. So first, in brief, introduction to DNA double-strand breaks. How are they formed? Why are they important? Why did they inspire us to work on these machines? Well, ionizing radiation and recombination nucleases are the natural sources of DNA double-strand breaks. In ionizing radiation, a particle or a photon moves along a defined track, transfers energy to water and other molecules that it encounters, and introduces chemical damage on the nanoscale in a very structured way. So that a very small amount of energy can do a lot of biological damage. And here's an illustration of a track intersecting a molecule of DNA and introducing clusters of damage on the two strands of the DNA that will result in a physical interruption of this chromosome. So most kinds of DNA damage the body handles quite well. All of you at this moment, there's a lot of DNA damage and repair that's going on in your body, 100,000 damage events per day, lots of metabolites of DNA excreted in everyone's urine. Double-strand breaks are different because they affect not just one gene. And after all, we have 2 copies of most genes, and most of the genome doesn't do anything anyway. But a double-strand break potentially affects thousands of genes, everything that's distal to that break on the chromosome. So cells really have an elaborate way of dealing with these breaks. They're gravely dangerous. A few dozen breaks are all it takes to kill a cell. This is a minute amount of energy being transferred, but it's being transferred in a structured way. So how do cells respond? Well, they have ways of tolerating this damage. Breaks are mostly dangerous when the cell divides. So cells have an elaborate mechanism for sensing when these breaks occur and going into cell cycle arrest. Cells can die. Now, clinically, this is very important. That's how radiation therapy works, is kills cells by introducing DNA double-strand breaks. Or they can repair them. And there are 2 pathways for repairing them, and I'm going to talk first about the simpler of these 2 pathways, which is something called non-homologous end joining. This is a machine that only has 6 components, and if you imagine a strand of DNA that has just encountered a break, it undergoes chromatin modification. There's a modified histone called gamma H2AX, and there's deposition of sensors and transducer proteins, of which a protein called 53BP1 is an example. I think it would be helpful to have a pointer with a battery that's a little more active. First step, we believe, in this machine is, thank you, is the binding of a protein called the Ku protein to the double-strand break. This is followed by the binding of the DNA-dependent protein kinase, the 3rd polypeptide. It's one of the largest proteins in the cell. Undergoes a very complex set of autophosphorylation events that remodel this complex. Recruits accessory factors, and importantly, recruits a complex of 3 proteins that has DNA ligase activity and that reseals the break. So the first example that I want to give of what we're doing with this machine is modifying the components so that they can be tracked optically. So the neat thing about this repair process, and the reason that we were so interested in pursuing it, it's a focal process. In the light microscope, you can actually see these repair foci being formed and disappearing on a time scale of a few minutes. So we have 3 ways that we're tagging the proteins. First is quite a conventional way, which is a genetic fusion of individual components to green fluorescent, cyan fluorescent, yellow fluorescent, or red fluorescent proteins. We've done that, we've used those. The second 2 ways are more advanced, and they involve semi-synthetic approaches, one of which is putting small tags in the components that can be chemically labeled either ex vivo or inside living cells in various ways. And the third, which I think in the end will be the most promising, is the most exotic, is actually putting a nanocrystal on these proteins, a quantum dot. It's very stable, very bright probes. This is an example. This is a piece of DNA. That's a quantum dot. And then when these are mixed with a protein intermediate that binds the DNA on the one hand and the quantum dot on the other, you see these complexes of intermediate mobility, which we've not only now formed in the test tube and are not yet in cells. To study the assembly, why would we want to study this assembly and disassembly? Well, it's a nice thing to study. It's an important biological process. But perhaps more importantly, as we go to modify the function of these machines, we want to be able to see what we're doing, see how we've engineered them, see the result of what we've done. we need to induce breaks in a controlled way. We have several ways of doing this. One is enzymatic, but I just wanted to show you something that we've invented together with Dr. Janata at Georgia Tech. When we irradiate cells, that's normally done in a $500,000 machine that has a lot of cesium in it and that requires a big fence around it and a security guards and health physicists to run it. It's very wasteful, right? You're irradiating all this air and all these culture dishes and all these places where the cells aren't. And we've developed a microelectrode. It's only 25 microns across, a little bit bigger than a cell. It's coated with a thin layer of isotope. The energy of that isotope is chosen so that the particles penetrate just a few microns beyond the end of the tip and eventually are arrested by the culture dish. And we can irradiate single cells one at a time. And using our bright fluorescent probes, we can collect the real-time assembly data. And so this is a 1-hour time scale, repeated images of the same cell that has a microirradiator positioned just above it. And you can see these repair foci being formed. Also, on a longer time scale, you can see them disappear. So we have now collected many gigabytes of optical data, and we are in the process of analyzing this data to look at the kinetics of assembly and disassembly. But our goal is not just to look at these complexes at a level of this chromatin indicator, but also to see the repair proteins themselves that are present in single copies in living cells. So this is a goal we've not yet achieved, but we hope to achieve with our quantum dot-ready proteins that we've made and are in the process of conjugating. It is going to require advanced microscopy. Optical microscopy is ordinarily limited by diffraction to about 250 nanometers in the XY dimension. Usually it's more like 750 in the Z dimension. We are applying new instrumentation with high sensitivity cameras, noise reduction algorithms, and a technique called structured illumination, where multiple images are collecting using patterned illumination light and combined. This is just an example early in the testing of this microscope. It's a conventional microscope image of some subnuclear structures, and this is with structured illumination. You can see the much better resolution, and in this short movie that just played, you can see this resolved in 3 dimensions. So we're very hopeful about the promise of these technologies. I do want to talk, though, about a clinically relevant example of how we can modify this machine. And this has to do with modifying the response to radiation therapy. So there are something like 1 million patients a year who will receive radiation therapy in North America. It's a very mature technology. Sometimes radiation therapy is given to prevent the recurrence of a cancer. Like early stage breast cancer, it's excised and the woman receives radiation in the hope that that will reduce the risk of recurrence and death. But a lot of times radiation therapy is given for cancer control. An example would be lung cancer. Most people that have lung cancer, particularly when it's advanced to a certain stage, will receive radiation as part of their treatment. It clearly extends life, reduces some of the symptoms that are associated with the cancer, but it does not cure people. The problem is that radiation there's not enough therapeutic gain. And we just heard from Dr. Blumberg about this problem. The war on cancer is a war on ourselves. There's some gain from radiation therapy. That's because tumor cells lack, they're dumb, they go on dividing in the presence of DNA damage, and they tend to, they're more susceptible to radiation injury, but not that much more susceptible. They replicate their unrepaired DNA, they divide, and they undergo post-mitotic cell death. So the idea that we had is if we could tune the double-strand break repair machine, if we could delay the process, you wouldn't have to completely inhibit it. You just have to delay it long enough for this competing process of cell division to happen in the tumor, and you would increase the therapeutic gain that's there naturally from radiation therapy. And of course, if you could target the therapy, that would improve it. If you could physically target it, it would improve it further. I think that would be true of any cancer therapy. If you could physically target it, you would improve it further. Here's the concept in cartoon form. Remember, there were 6 polypeptides that assembled. And what we have is we have an antibody fragment that binds right in the middle of the third polypeptide and prevents those phosphorylation events and prevents the further assembly of the complex, and you're left with a poisoned incomplete repair reaction. Stephen Lee talked about single-chain antibodies yesterday, introduced them. They're 3 to 4 nanometers in size. They're produced recombinantly. We happen to be working with one that has very high affinity for its target, which has been precisely mapped in a conserved regulatory sequence in the center of this key enzyme in the repair pathway. So antibody therapy is very well accepted for cancer treatment, but all the therapies we have work on the outside of cells, and I'm proposing to use this really nice specific platform, but on the inside of cells. So how would you deliver the antibodies? Well, we proved the concept by micro-injecting them into cells, and we observed a lot of sensitization to clinically relevant radiation doses. Microinjection is nice. It may have applications to other clinical problems, although clearly not to cancer, because of the precise volume and timing of the introduction of the protein. You can use unmodified proteins. You don't need to modify these in any way with carriers, and you have control cells that are uninjected on the same plate. And my colleague, Dr. Shu-Yi Lee, talked quite a bit about these proof of concept studies last year. But today I want to talk about a more practical delivery method, which is receptor-mediated endocytosis. And this allows for cell-specific targeting. It's a fairly mature technology. It's been used for other things. And importantly, it offers a path for clinical translation. And in particular, we're using folate receptor-mediated delivery. Folate receptor is overexpressed in many cancers. Folate's a cofactor for several of the reactions of DNA synthesis, and cancers do a lot of DNA synthesis. Binding to the ligand promotes non-destructive internalization and release of cargo, proven for model proteins, and we know that it would be translatable to the clinic if we could get it to work. We coupled the single-chain antibody to folate through a reversible disulfide bond. We made this in 2 flavors, and in one case, we also incorporated an endosome disruptor peptide, but we found really that these 2 were equivalent, and the simpler one was easier to make. We confirmed the conjugation in several ways, by spectroscopy, by gel electrophoresis, and we showed that there was minimal or no interference with the specificity of epitope recognition. And here's the thing, it works. So this is the key piece of radiobiological data. This is a clonogenic survival curve in vitro with cancer cells. We've done it with several lines. I'm showing just one. A typical survival curve with a shoulder in the low dose range and then increasing sensitivity as you increase. And clinical radiotherapy is done through multiple fractions of radiation right here in the shoulder range. And so the goal of a clinical radiosensitizer would be to eliminate the shoulder in the cancer cells. And this treatment does. If you compare control treated with experimentally treated, there are ways to quantify this. You take the integral under these curves from 0 to infinity. You come up with a sort of a figure of merit for the treatment. And this is 1.92. Well, you can think of it as a reduction in the dose needed to get a given effect. And that's considered a good figure for a lead compound. It would be something you definitely want to pursue. We know a little bit about the mechanism. We know it does block one of these autophosphorylation events. This is a phosphoepitope that's seen in the DNA-dependent protein kinase by immunostaining in individual cells, and in the treated cells, that phosphoepitope is not detected. So where does this stand? The radiobiology in vitro looks promising. There are various issues with engineering the antibody fragment, with its stability, with modifying it, how you deliver it. That's all underway. I'd like to be able to show you live cell imaging of the uptake and fate, as well as animal experiments, and those are in progress. We hope to have those complete within the next year. And importantly, we've established a discovery paradigm that I think can be extended to other examples of things that one might want to do. We picked a disease, which was cancer. We had a device, which is a single-chain antibody that was modified through the attachment of a nuclear localization sequence and folate and some other things, and we had a delivery method. And in principle, all 3 of these are independent, and you could choose another disease, another device, another delivery method. So I want to speak very briefly about this idea of re-engineering for new function. And I want to tell you that nature's done this with this machine, with the one we're working with. There is a natural example of this having been re-engineered. So DNA repair is of course, nature's only way to alter gene sequences. And non-homologous end joining turns out to be one of these very ancient functions that you find in many organisms in different domains of life. This Q protein has very ancient homologs. So this was around. Right? This problem of double-strand break repair has existed since the beginning of biology, or since the beginning of the DNA world, whenever that came in the progress of biology. And then about 400 million years ago, something happened. Probably a transposable element got integrated into the common ancestor of the jawed vertebrates. And that transposable element encodes a protein complex, 2 polypeptides, recombination activator genes 1 and 2. And all these do is incise the precursor genes of antigen receptors. And those precursor genes already existed in organisms like the lamprey, but they couldn't rearrange to form new receptors, to recognize things that you'd never seen before. And all these proteins do is they make a cut, and then they retain the ends, and they shepherd them into a repair pathway that was pre-existing. Our goal is to emulate that process, to make a cut in a gene of interest, and then to shepherd that into a repair pathway of our choosing. And at that point, we begin to diverge from this example of the immune system, because first of all, we like a gene that would be useful to modify. And well, we thought we'd cure cancer with this, right? We just inactivate the oncogenes. And people told us, well, that was, maybe it's not the place to start. Maybe we should start with single gene disorders. And we are told that there are about 5,000 of these single gene disorders, some of which affect just a few patients worldwide, and some of which are quite prevalent. And in Dr. Bloomberg's talk, he actually mentioned one of these, which is sickle cell anemia. The interesting thing about this is that this same mutation has recurred 4 times independently in human evolution. So all patients have the same mutation. There are about 90,000 patients in the US, many more worldwide. It is a bad disease. You'd definitely like to cure it. It's monogenic. There's a good animal model, and there's an accessible stem cell. So it has a lot of the characteristics of a good place to start. Our device will involve incising the sickle cell gene. In this case, we'll use synthetic nucleases. We thought of using the RAG endonucleases, but they're not structurally well understood, and it just wasn't, we thought, the way to go. So instead, we're going to use zinc finger nucleases that we already have. And we're exploring delivery by transferrin receptor-mediated endocytosis. The idea is that we'll create a double-strand break near the sickle cell mutation, or in principle any mutation for a monogenic disease. We will engage a specific repair pathway. In this case, it will not be non-homologous end joining. It will be the other double-strand break repair pathway that involves template-dependent synthesis, initiated by binding not of the Q protein, but of a protein called RAD51. And that filament then invades a donor template. And so this is now a modification of a machine that does not involve adding 1 component, but adding 2, at minimum, the zinc finger nuclease and the donor template. Where are we with this? We have the zinc finger nucleases for globin and also for some model genes. We are working on transferrin receptor-mediated endocytosis. The challenges are efficiency and specificity. We'll use our real-time visualization capability. We need to be able to monitor and suppress mutations and rearrangements that might be introduced inadvertently. And an ideal gene correction machine would be independent of foreign DNA and proteins, non-immunogenic, and amenable to temporal control. We are exploring ideas for photo control of the process. I want to acknowledge my collaborators, Gang Bao at Georgia Tech and the other members of our center, as well as the people in my own laboratory that are working on this, and tell you that I've covered 4 topics: double-strand breaks and the response, imaging that response through modified components of the machine, inhibiting the response by adding 1 additional component, and the idea of re-engineering the repair machine by adding 2 components, a nuclease and a donor template that will shepherd this into a recombination pathway. Thank you very much.
Thank you for that fascinating presentation. Are there some questions, please? Your name and
Oh, I'm Tad Hogue. You mentioned the second mechanism of DNA repair uses a template. So in the normal use of that, where does that template come from?
It could be the homologous gene on the other chromosome, or it could be a sister chromatid. These two mechanisms work in sort of a yin-yang way. The non-homologous end joining is an acute response to things like a radiation injury. The homologous recombination seems to be more important in genome maintenance because people with, well, BRCA2 patients would be an example of people with a sort of a subtle defect in that makes them susceptible to genetic instability and cancer.
question?
Kester, Penn State, excellent talk. So one of the double-edged swords of radiobiology is it does kill the cancer, but it also kills the endothelial cells, and you therefore have trouble adding secondary drugs, or in your case, transferrin-mediated small fragments of antibodies. Is there some way you can take your technology and gear it to a targeting approach so that the transferrin receptor-mediated antibodies only go to the cancer cells and not the endothelial cells of the nascent vasculature?
Well, I don't know, maybe it's a triple-edged sword.
Yeah.
So sometimes you want to kill those endothelial cells. I've talked about
Right.
this as if you're killing the tumor cells, but in
Right.
fact, there's some debate whether, it's the tumor cells or some of the other cells that are associated with the tumor. Assuming that we're killing the cancer, it is not clear at all that normal tissue damage and killing the tumor work by the same pathways. In particular, killing the normal cells is probably not working by post-mitotic cell death because the cells either aren't dividing in the first place or they have intact cell cycle checkpoints. So I think you can make an argument that radiation works anyway and that we are exploiting the natural differential and just increasing the differential. And then, of course, with any nano approach, there is also the possibility of adding a layer of targeting. So I gave as an example the folate receptor because it's used in tumors, but I think we can imagine a lot of other receptors that you might want to target for your favorite cancer. I'd also like to say we think this endocytosis of the free protein is going to carry us at least as far as some of the preclinical studies. But clearly there's a lot happening with nanoparticle delivery, as you told us yesterday. And so I don't know that that'll be the final device. I think there's plenty of room at the bottom to develop other devices.
I'd like to make a comment. You referred to the early genomes in trying to explain the big mutational change that took place. One of the things that's really possible now, because of the assembly of so many genomes in one place in a database, is kind of comparisons, ready comparisons of genes. And if we want to look at other functions of our genes, a very useful thing to do is look at the homologous genes in very early species. Because many of those effects that were useful to the earliest species in the evolutionary story are actually still present in our genes, and probably some modified form. So a systematic study of genes it's now available. You can look at them and see what they are.
So I have to tell you why it wasn't obvious that this system was present in other organisms. Some organisms have lost it. And in particular, E. coli has lost this whole system. It gets by with other repair pathways. And for a long time, at least somebody like me, maybe thinks E. coli is the only bacteria that exists, and it didn't have it, and so it must be specific to higher organisms. Well, it turns out lots of other eubacteria have the pathway. I am not sure whether it's turned up in the archaeal genomes yet or not, but the prediction is that it would, because it's clearly ancient.
One of the things that we encouraged in the astrobiology program was the study of extremophiles. And I think it really helped move the field forward a good deal, and many more are now unknown and understood.
So we've passed the 1,000 genome barrier, and I guess we're someday maybe going to reach the 10,000 genome
I'm sure that'll
level.
happen.
Most of these are single-celled microorganisms.
Well, thanks again for an excellent talk, very stimulating, and we'll go on to our next speaker.